Well, welcome to everybody. I know some of you are joining us straight from your living rooms across New Zealand, so this is wonderful. We have some of our LVC staff throughout the offices watching as well, and others. So welcome to our winter-spring webinar series. My name is Matt Eby. I'm a Regional Manager of Support Services with Leukemia and Blood Cancer New Zealand. I am joined by Dr. Rob Weinkove, and I just want to maybe just give a little brief intro for him before I turn it over to Rob. So Dr. Rob Weinkove is a hematologist at Wellington Blood Cancer Centre and clinical director at the Malaghan Institute of Medical Research. After studying medicine at the University of Cambridge and King's College, London, he trained in hematology in London and Germany and undertook a PhD in immunology at the University of Otago. Rob's research interests include B-cell malignancies, immunology, and supportive care. Rob leads a chimeric antigen receptor, Car T Cell development program at the Malaghan Institute, and is the principal investigator for Enable, which is New Zealand's phase 1 Car T Cell trial. So with that, very happy to have you along today, Rob. Really appreciate it. And the talk titled Updates on Car T Cell Therapy in New Zealand. And one last thing for all of our viewers, yep, there's a few of you in the room. You'll see the Q&A box. Feel free, as Rob's talking, you can type your questions right into it, and afterwards we'll be able to field the questions and hopefully get some answers. So once again, over to you, Rob. Thank you.
Thanks very much, Matt, and thanks for the invitation to speak today. As Matt said, I've spent the last few years during my the research part of my role, establishing manufacturing and processes for a clinical CAR T cell trial, which has been quite a big deal that we've been embarking on. Took a lot bit more work than we thought it would, as these things usually do, but we are currently running a phase 1 CAR T cell trial and have been recruiting some patients from around the country. So I'll speak to that in a moment. I'm just going to share a screen with a presentation and some slides. Hopefully you'll be able to see me in the corner. I'll let Matt's doing his wizardry with the arrangements for the webinar. So as Matt said, I work part-time as a hematologist, so 2.5 days a week as a hematologist at Wellington Blood Cancer Centre at Wellington Hospital, and I'm clinical director at the Malaghan Institute. And the current role I'm principally involved in is as principal investigator for the Enable trial, which is our phase 1 CAR T cell trial. And thanks, by the way, in case I forget later, to Leukemia and Blood Cancer New Zealand, which has been providing some support both for one of our PhD students and also for the travel of some patients to access this trial. So I'll just advance the slide. So I think for many of you probably say a great deal about lymphoma in general, but I always found it confusing as a medical student. What's the difference between a lymphoma and a leukemia? And in some respects, we make things complicated for ourselves as hematologists by subdividing all the conditions into many different subtypes and giving them all slightly different and often very complicated names. But the reality is that lymphomas and leukemias, a fraction of those are cancers of a type of cell in our body called B cells. And these are the cells that normally make our antibodies. So they're often present in our bone marrow, which is where the B comes from, but also in our lymph glands. And they're responsible for making the antibodies that protect us against disease. And cancers of B cells are called lymphomas if they're mainly in the lymph glands or mainly forming solid tumor masses. And they're called leukemias if they're predominantly in the blood and the bone marrow. In some cases, the condition can sort of vary between a lymphoma and a leukemia. An example of that is chronic lymphocytic leukemia and small lymphocytic lymphoma. They're two different names, but they're basically the same disease. But as I say, we subdivide these conditions and it makes it rather complicated. But if you just take the cancers of B cells, the B cell lymphomas and leukemias, overall those alone are the 6th most common cancer in New Zealand. And around 1,000 people each year are diagnosed with B cell leukemias or lymphomas. And so this is a significant need, and there's lots of people, including some of yourself, I presume, that have these diagnoses. The good news is that for the slower growing ones, they may not require treatment at first at all. And most people respond really well to our initial treatments. And in fact, some people are cured, particularly with the more aggressive lymphomas like diffuse large B-cell lymphoma, are cured with our first-line treatment. So we have really good treatments early on for these types of malignancies. We use chemotherapies, we use antibody treatments, and increasingly we use tablet medications for some of them. So we do have good treatment options. However, we still struggle, and we still struggle because in some people, the condition becomes refractory to other treatments, so it doesn't respond at all in the first instance, or in some people, it keeps coming back multiple times after other treatments. And then we end up with a situation of having relapsed or refractory disease. And that's where we struggle, because by the very nature of that, people have often been through our conventional earlier treatments. And the issue is that the population of the lymphoma or leukemia cells that remain are the ones that are relatively more resistant to the treatments that we have available. And this represents a key area of need in this type of condition. And we know as hematologists, and we're often, we like to be optimists, we all like to be optimists, we know as hematologists that the immune system, T cells, can actually fight B cells. So our T cells are cytotoxic cells of our immune system. They're the cells that typically we go around fighting virally infected cells or cells that are on the cusp of becoming malignant. We can actually use one part of the immune system to fight another. We can use T cells to fight malignant B cells. And we know that through a long history of doing bone marrow transplantation, which is a procedure where we give someone an entirely new immune system, entirely new T cells. And sometimes that works really well. This PET scan, it's a rotating image, is of a patient I treated when I was training in the UK. You can see some hot areas, which are the areas that are dark on that scan. Some of those are completely normal. The brain at the top is always PET avid. The heart always takes up this radio labeled glucose. But what there is sort of midway down into his belly is an irregularly shaped mass. Not the bladder, which is very dark and is beneath, but a irregularly shaped mass, which is a mass of lymphoma cells in the what we call the retroperitoneum, so an area sort of packed around the sides of the kidneys, or beneath the kidneys. You can see the kidneys actually lit up there. And we gave this patient a transplant, and he had the immune suppression, as we often do with this type of bone marrow transplant, and did a scan, another PET scan quite early, and in fact the tumor mass had actually got larger and denser, so the actual tumor had grown early after the bone marrow transplant. So to try and get the patient's new bone marrow to fight the lymphoma in this case, and this is a mantle cell lymphoma, we withdrew the immune suppression. The patient developed an immune reaction called graft versus host disease, fortunately quite mild. And another PET scan, the tumor had shrunk dramatically, and at a later PET scan, the tumor had gone away completely. Now, this, of course, does not always happen, but when it does, it's great. And this patient actually remained in remission 10 years later on. But the problem, and this is one of the problems with doing allogeneic bone marrow transplantation, is that he had symptoms, had problems despite being in remission. So he got rid of the lymphoma, but he had chronic graft versus host disease, was going on and off courses of steroids to manage this immune phenomenon. Because of the immune suppression, he was getting various infections. So the bone marrow transplantation did what we wanted it to in terms of eradicating his lymphoma. But the downside was, by giving this whole new immune system, we'd thrown away all the rest of his immune system, and we created a range of sort of immune type problems that carried on causing symptoms for a long time. This doesn't happen to everyone after bone marrow transplantation. Some people don't get this at all, but it's one of the issues that we have. It's a little bit like rolling the dice. It can work fantastically well, but it can have these side effects, and we can't predict with accuracy who's going to get those side effects and who's going to fare well. So one of the challenges that I've always been intrigued by is how can we use the immune system in a more targeted way, perhaps, so get the same kind of effect against the tumor, but without causing the less predictable side effects. And many of you might have seen the stories that came out around 2012 and the Obama era. It seems like a dim and distant past, where we had stories, particularly early on, from a girl called Emma Whitehead, who became literally the poster child for this new type of treatment called chimeric antigen receptor T cells. And she was treated for a B-cell acute lymphoblastic leukemia after exhausting all of the treatments and went into complete remission. I'll talk in a moment about what these these cells are and how we make them. So you may have also heard more recently some stories in New Zealand in the press. So David Downs has very publicly talked and blogged about his experience of lymphoma, and then went overseas to the United States and received a type of CAR T cell therapy that put his lymphoma into remission very successfully. And there he is pictured with his family. These are public domain stories. And you may also have heard in the last year that this is starting to become a standard of care type of treatment in Australia, where initially the Peter MacCallum Cancer Centre is the first treatment centre, but there are other centres opening up in other states in Australia to deliver this type of therapy, CAR T cells, as a routine for highly selected patients with certain types of relapsed and refractory lymphomas and B-cell leukemias. So what is this technology, and with all this information and this really exciting field, how do I feel that we could try and bring this here and try and make this something that we could make more available? So this picture on the top left is a sketch of the chimera, an ancient Greek beast, partly one animal and partly another. And that's where the term chimeric comes from, because actually, essentially what you're doing with chimeric antigen receptor T cells is you're sort of hacking the immune system. What you do with CAR T cells is you take, and I'm not going to go into this in too much detail, but you take part of an antibody, and we actually already use antibodies to treat lymphomas, so antibodies that latch onto the B cells. It's a little fighting fire with fire, because we're using an antibody-based treatment to fight a cancer of an antibody-producing cell, which is kind of intriguing from an immune system perspective. But a chimeric antigen receptor, so we take the part that recognizes a tumor cell from an antibody, and we attach it to part of the T cell receptor. We create this new synthetic chimeric receptor, a new protein that effectively pastes together parts of other proteins to achieve the goal of allowing a cell now to recognize, very specifically to recognize a particular cancer cell. And these CAR T cells have been through various generations over the years. First generation CAR T cells actually developed a long time ago in the early 1990s and latched onto the target but didn't really kill tumor cells. Second generation CAR T cells actually also first were developed in the 90s. And now what we're working on is a so-called third generation CAR T cell, where we try and boost the effect of this. And I'll talk about that again in a moment. And there are several CAR T cells that are either licensed in some countries or in advanced clinical development. One of them is called Kymriah. That's the one that's been made available for highly selected people in Australia. It's made by Novartis. Its long name is tisagenlecleucel. Yescarta is made by Gilead, and that's licensed in the US and Europe. It's also called axicabtagene ciloleucel, and Celgene also have a product called Lisocell. These are all directed against lymphomas, and there's also investigational CAR T cells for myeloma, as some of you may well be aware already. So this is a cartoon that we made up at the Malaghan Institute just to illustrate CAR T cell therapy and what it involves for patients. And I'll try and just step through that. And for some of you may have had a type of bone marrow transplant called an autologous stem cell transplant. And from a patient perspective, some of these steps are very similar. So we start off with a patient, and the first thing we do is we have to take some patient T cells, some patient immune cells, and we get those from the blood. And we get them from the blood by a procedure called leukapheresis. That's done, we're doing that at the New Zealand Blood Service. And it's the same procedure as that's used to gather stem cells for a bone marrow transplant. It's also used by people who donate, some people who donate platelets and plasma at the New Zealand Blood Service. And effectively what happens is you put your arms out to have a line put in each arm, and this machine takes blood off one side, centrifuges it to separate out the white blood cells of interest, and then returns the rest of the blood components, the plasma, the platelets, the red cells, back to the patient. And it effectively then processes over a few hours, a couple of volumes of blood, and we end up with a reasonably enriched population of immune system cells that we can take off to the laboratory and work on. We then have to purify those a little bit further in the lab, and we've established CAR T cell manufacturing at a small scale with MedSafe approval here at the Malaghan Institute in Wellington. So what we do is purify the T cells further with a number of additional washing and purification steps. We actually use some magnetic beads that latch onto the T cells and allow us to purify them to a high extent, make them about 99% pure. And then what we have to do is we have to take those T cells and we have to then get them to express, to have on the surface of them the new receptor, the chimeric antigen receptor. And to do this, we reprogram the T cells by introducing a new gene into them. And for that purpose, like the commercial CAR T cell therapies, we use something called a retrovirus. And these are viruses that are capable of injecting their DNA into a cell. Now, what it's not is it's not a functional virus. It's not a virus that can actually cause harm. It's a sort of a highly modified lentiviral vector that in practice is only capable of delivering its payload of the new gene, the gene that encodes the chimeric antigen receptor, and then it can't reproduce itself. So it's what's called a replication incompetent virus. And there's a whole bunch of work we had to do to establish manufacturing of that vector and to do a great deal of safety testing on that and gain a great deal of approval work. What we end up with then is some T cells now that have been reprogrammed, and they're now capable of expressing on their surface this new receptor that enables them to specifically recognize, in this case, B cells, so recognize the tumor cells. And when they do so, they can kill those tumor cells. We expand the T cells up to large numbers in the laboratory over about 10 days by culturing them in a specialized facility. And then we have to freeze them down. We have to do a bunch of safety testing on the CAR T cells to make sure they're what we expect they are, to make sure they don't have any contaminants and so on. And while we do that, we start to arrange for the patient to come back to receive the CAR T cells. Now, if we just gave the CAR T cells directly into the blood, they wouldn't do very much. They might attack some lymphoma cells, but they wouldn't proliferate or expand within the patient a great deal. So what we actually do is there is what's called a conditioning chemotherapy. It's 3 days of relatively mild chemotherapy. It's quite a lot milder than the chemotherapy we use for a routine bone marrow transplant. But the patient gets 3 days of outpatient chemotherapy. We usually do that on Wednesday, Thursday, Friday. No treatment over the weekend. And on the Monday, we give the CAR T cells into the blood. And by giving this conditioning treatment, what happens is that the CAR T cells are then able to expand inside the patient. And so unlike any drug, the actual cells will expand inside the patient in reasonably large numbers over 2 or 3 weeks, hopefully recognize the lymphoma cells, attack the lymphoma cells, and once there are no lymphoma cells left, the CAR T cells then reduce in number again. Now, they may persist later on for a variable period of time and help prevent lymphoma relapse. There's a period where we have to monitor patients quite closely, and that's because during this period of the CAR T cells latching on and recognizing the tumor cells, this can cause an inflammatory-like reaction. So people can get a phenomenon called cytokine release syndrome, which is fevers and low blood pressure. It looks a bit like getting a bad sepsis or a bad bacterial infection. And we have to monitor very closely for that. We also monitor because there are other risks such as confusion or seizures in the most extreme cases. So we monitor closely for several weeks after the CAR T cells have been administered in case these toxicities arise. We have one of the pieces of work we had to do for this trial was develop some specific protocols for recognizing those problems and how to treat them. So that's been very useful to set that up. And just overall, and this is a fairly broad brush approach to what the response rates are like. And I just want to point out that I'm talking here about response rates of licensed or advanced investigational CAR T cell therapies. There's very many different ways of making CAR T cells. These are not data for our CAR T cells that we're manufacturing here, which are still only being given to a few people so far, and we don't have a great deal of data on these yet. So for relapsed B-cell lymphomas with CAR T-cell therapies, and this is generally people who've exhausted other treatment options, around about a 50% complete response rate. And most of those who do have a complete response seem to still be free of the lymphoma 2 years later. What we don't have yet is very long-term data to know whether those people might be cured or not. But we're optimistic that the relapse rate seems to be very low beyond a year or so after the treatment. In acute lymphoblastic leukemia, a rarer condition, the response rates can be really high, 90 to 100%, and about half are still free of relapse 18 months later. So that looks quite promising too. And there's an investigational CAR T cells now for myeloma, and the response rate also seems to be very high, but what's much less certain is whether people actually stay in remission long-term or not with the current CAR T cells for myeloma. But one of the intriguing things about CAR T cells is that because it's very much a designer therapy, you can make CAR T cells directed against almost anything you want to. So there's a huge number of different types of CAR T cells in development and a huge amount of work internationally to try and modify this treatment and make it more effective, make it useful against other types of cancer. And I think it's really important that we in New Zealand can benefit from that and be part of these trials, and also hopefully that will accelerate the pathway towards having routine standard of care CAR T cell therapies. So I've sort of over the last few years, and I started this program in 2017, was asking, well, how can we develop, taking two views, I suppose, a global view and then a New Zealand view. So the global view would be how can we develop more effective and safer CAR T cell therapies? And the New Zealand view is how can we secure availability of CAR T cell therapies here in this country? And so what we want to see internationally, and they do hang together really because we're part of a global community. If we could set up our own CAR T cells, but if we can't get some sort of commercial engagement, it's unlikely to be able to deliver those in a large way. So the sort of things that we need are higher response rates. So what about the 50% of people that don't respond well to existing CAR T cells for lymphoma? We want a broader applicability. Can we use those for more types of lymphoma? Can we use them for more hematologic cancers? And with all of this, can we make sure we have an acceptable safety profile? Now, the safety profile, you know, a chance of severe toxicities of 1 in 5 or 1 in 10 might be acceptable for people who've exhausted all of the treatment options, but maybe we could be using CAR T cells in a larger fraction of people if we had safer CAR T cells. So can we do that and make them safe? And how can we secure availability of CAR T cells in New Zealand? Well, at the moment, the commercial CAR T cell therapies are incredibly expensive, around $300,000 or $400,000 US if you wanted to get them commercially. Plus, in the US, the cost of administering them, which can be at least as much again. We thought that in order to be able to do small-scale trials, we needed to have some onshore manufacturing capability, and I'll speak to that later. It would be great to have national access to trials, and what people want to see, the international large companies that run clinical trials want to, they're looking for trial centers, but they want centers with CAR T cell treating experience. They want centers with experience of managing the toxicities. And so having that experience before is really important. We want our regulators to be aware of CAR T cells and try and work our way through the regulatory environment. The regulatory environment for medicines and gene technologies varies substantially from country to country. And one of my jobs over the last few years has been navigating that process. And in all of that, if we do develop new treatments, we want to protect that intellectual property and try and preserve New Zealand rights. So if we do have a wonderful success, try and make sure that we're able to deliver that type of treatment here in the future and it doesn't all just go offshore. So to do that, we've really gathered together a very global collaboration as part of this process. And we've had built together a team that have members from originally from Italy, from the US, from the UK, from China. And we've linked in with our CAR T cell treating clinician colleagues in Sydney and Melbourne. We are sending parts of our cell samples off to Auckland for testing. We have a data safety monitoring committee's members from Christchurch, Auckland, Dunedin, Melbourne, Washington in the US. And we've really sort of forged very much a global collaboration to try and make sure that what we're doing is right, because this is a new type of therapy for New Zealand, and we don't operate in isolation from the rest of the world. So we were quite pleased last year to get most of our approvals in place, and it was quite a long mission. So at the top left is one of our healthy donors. So before we could start manufacturing CAR T cells and running a trial, we had to validate that we could do this. So we actually had 4 healthy donors. This is one of our nurses who kindly volunteered to donate her cells, and there she is attached to a leukapheresis machine, taking off some cells. And at the bottom left is our manufacturing laboratory at the Malaghan Institute, where the cells are being manufactured with a couple of team members. And it's the type of laboratory we actually manufacture the cells in is actually very simple, in essence, by regular laboratory standards. But what's unique about it is the particular procedures and policies in place and the airflow handling. So there's 2 antechambers before you can get into that laboratory. The big silver thing that looks a bit like an oven on the right is a sterile pass-through hatch to allow things to pass through from the antechamber into the room and be kept sterile throughout. And the reason that they're wearing all that garb is that's an incredibly sterile environment. So we have to have fewer than 20 particles per cubic meter of air, which is a tiny level of cells that's around 10,000 times cleaner than the air that you or I are breathing right now. And that's because we're going to be handling patient cells. We don't add extraneous antibiotics and things, so we can't afford any bacteria or fungal spores to get into those cells, and we're going to be delivering those cells back to people who are immune compromised. So absolutely critical that we do all that. And a lot of the work is around the policies and procedures for this. There was a lot of work for the GMP laboratory to get their MedSafe license. So MedSafe in New Zealand is the body that regulates medicines. And effectively what we're doing for each CAR T cell dose is we're manufacturing an individual patient's medicine. So there's an extensive record of that medical batch. We had to get approval from a committee called the Gene Technology Advisory Committee, which regulates trials of gene therapies in New Zealand. We had to get approval, of course, from the Ethics Committee, and approval in this case because the CAR T cells themselves in New Zealand legislation are classed as a genetically modified organism. We had to get an approval from the Environmental Protection Agency, and that was a bit of a challenge, really, because the New Zealand GMO rules were set up for seeds or animals. In fact, CAR T cells are not, they're not a self-sustaining organism. In fact, they're not an organism. They can only survive within the intended recipient. But nonetheless, we had to jump through a number of hoops to get them through that approval. But we did manage to succeed that. It took us longer than we hoped, but we did so, I think, in fairly good time overall, considering the amount of work that was put in. And last year we gained our license to manufacture genetically modified T cells, including the ability to manufacture the vector to make the CAR T cells, and including the CAR T cells themselves. And our CAR T cell product that we're using in our current trial is called WZTL002. It's Wellington Therapies Limited 2. And the trial that we're running is called Enable. It's always good when you're running a trial to have a sort of catchy title for it. And we called it Enable. It stands for Enhancing CAR T Cell Therapy for B-cell lymphomas by engaging Toll-like receptor 2 or TLR2. And clinicians around the country are aware of this trial and its inclusion-exclusion criteria. It's registered on the online international registries, and it's also on a New Zealand app called ClinTrial Refer, which carries details of clinical trials open across the country. Now, it's not the same. We're not doing the same kind of CAR T cell therapy as some of the existing products. We have to make sure that this is scientifically important as well. And so if you took Yescarta's CAR T cell product, which is really the product with the highest response rates for B-cell lymphomas, that has inside the cell a couple of domains, CD28 and CD3 zeta. Don't worry about what those are. And our CAR T cell product has one extra domain. It's got a Toll-like receptor 2 domain. And without getting into the science, we have rationale to believe adding that domain may make the T cell activity against the lymphoma stronger. And so ours is what's called a 3rd generation CAR T cell. It is a phase 1 trial, which means it's a first-in-human trial. And it's a trial where we're looking at the safety of this product, and we are trying to find out the right dose of this product. So we've just completed treatment of the first 3 people at the lowest dose level, and we will be moving on hopefully next week to start treating the 4th participant at the next dose level. So we treat 3 people, we assess toxicity, then we go and treat increasing the dose until we find an effective dose, and then we hope to do additional trials beyond that. So we do not yet know the safety and efficacy of these CAR T cells. Having said that, we have picked the CAR T cells because we have biologic reasoning to think they may work very well. So what are some of the things we're doing? Alongside this is a huge amount of laboratory work. And now Yasmin Nouri, who is a doctoral scholar, she started with us last year, and She received generously a scholarship that was administered by Leukemia Blood Cancer New Zealand, the John Waller Doctoral Scholar. And one of the pieces of work that Yasmin's been doing in our lab is working on some methods of assessing the CAR T cells and assessing their efficacy in the laboratory. And she's working on a system of live cell imaging. And this image, our model cancer cells are a fluorescent green, and our dead and dying cells light up in a red color. The CAR T cells themselves are actually not labeled, so they're sort of in the background. You can see them on the left picture as gray. And on the right are a couple of videos. The top one, which I'll get started in a moment, the top one has some cancer cells that don't express the B cell marker, and the bottom one has some cancer cells that do express the B cell marker that we're targeting. And hopefully those will start now. And you can see that over a couple of days in the top video, the cancer cells that don't express the marker that our CAR T cells can target continue to grow unchecked, and they take over the whole plate. You can see huge numbers of green cancer cells growing. In the bottom videos, and the video is just rotated around and started again. In the bottom video, and the time points are the same at each point, the cancer cells have the marker on the surface that B cells have, and they are being targeted by the CAR T cells. And what you can see what's happening is that the cancer cells are clumping together into these great big clumps, and you can see lots of bright red, which is the cancer cells dying. And so this, alongside a number of other assays, is one of the tests that we're using that's demonstrating that our CAR T cells are specifically killing cancer cells that express the target marker, but not killing other cells, which is important. Because what I want to do is kill the cancer cells without causing some of those potential for long-term toxicities that you can get with bone marrow transplantation. And so we do lots of things with lots and lots of planning for our CAR T cell trial. And just to emphasize again that this is first in human trial, and I can make no assertions at the moment, or no assertions, sorry, about the safety profile or the effectiveness of our cells, because we haven't treated enough patients yet to know. But what we do is that patients have to be seen and referred by their own clinician, because really, especially with such an early phase trial, we have to be very stringent about the inclusion criteria. We have to know that people are not at undue risk of toxicities. We try and get as much of that process of assessment done locally as possible, so we're not sending people across the country unnecessarily. We do a telephone consultation, we talk through things, we check the data information from the referring clinician. We then, if we think someone might be eligible, then we bring them to Wellington for a visit for consent and for some screening procedures. We have to check heart and lung function, we have to check kidney function, and do a range of other assessments locally. And then if they do appear to be eligible, we enroll them. And then we do that procedure called leukapheresis, where we get the cells that we're going to make CAR T cells from, and then patients go back to their own region while we try and manufacture CAR T cells. And we're still working through issues of manufacturing. We've had a couple of the patients on the trial so far, we've had to manufacture the cells more than once, and we're still refining our manufacturing processes to make them as effective and reproducible as possible. Once we've made an adequate CAR T cell dose and we think the patient's fit for treatment, and just bear in mind that we do allow for patients to have what's called bridging chemotherapy if their lymphoma, because it can be a couple of months between being enrolled into the trial and being treated, and so we do allow for people to have chemotherapy back at their domicile health board if needed to contain the lymphoma. Then we do a PET scan to evaluate the lymphoma, and then 3 days of fludarabine and cyclophosphamide chemotherapy. And essentially that's to reduce the number of normal T cells and allow the CAR T cells after they've been given to expand better inside the patient. 2 days later, we give the CAR T cells, the single infusion through the veins, and then we monitor very closely. And what we're doing at the moment is actually having people as an inpatient on the ward for the first 14 days, really as a precaution. And then if things are well, they go to a near hospital hotel and come back each day for an assessment on our day unit. And then beyond 3 weeks, we start to relax the frequency of those assessments. And we aim, if there aren't toxicities, to let people return home 28 days after the CAR T cells, and then come back 2 months, 3 months, and then 3 monthly for 2 years after that. So just to come to where we're up to and what I think we're doing to address what I think these issues are of safer and more effective CAR T cell therapies and accelerating really availability of CAR T cell treatments in New Zealand. So at the current time, we're doing this ENABLE phase 1 clinical trial, which is currently recruiting. We don't know how many patients we'll treat in this trial because it's a phase 1 trial. So if we get bad toxicities, we may have to pause the trial again. So it's not like this first trial is going to meet the clinical need. We don't know how effective or safe the CAR T cells are, but it is a start. And it's a start because it puts us in the scientific space of doing CAR T cell trials. It demonstrates that we can manufacture them. It overcomes we had to overcome many of the regulatory hurdles, and we think it'll be faster to overcome those for future CAR T cell therapies. And it also means we have had to establish systems for managing CAR T cell toxicities in New Zealand, which we think is really critical based on our discussions with international pharma companies to enable other larger scale CAR T cell trials here. So we have protocols and policies for the management of the various toxicities that we have put together in collaboration with our other clinicians, and we've had to use them and trial them out in our trials so far, and they've worked very well. We're also conducting some mechanistic research. This is part of the work that Yasmin is doing. We're looking at how this particular domain within our CAR T cell is working. We also are keeping close tabs at the moment on data for CAR T cells for other malignancies, including myeloma. We're a small trials unit at this point, and we can't run trials for every indication, but we do have collaborators who are working on CAR T cells for other indications, and we're watching that very closely. It may well be that our CAR T cell for lymphoma is no better than the existing CAR T cells, or even has more toxicities, but we may have other CAR T cell therapies that we can help develop and run in other trials in the future. And we are, through our work, trying when we can, if we're coming up with new ideas, to try and make sure that they're protected, and that we write contracts with funders that ensure we secure long-term New Zealand rights to those products. We're also working on some other more, futuristic ideas. We're looking on ways to improve CAR T cells' ability to kill other tumor cells in the neighboring vicinity of the ones that they target, which we think might be important for treating solid cancers in the future. We're looking on a mechanism for a new safety switch, and we got some grant applications and team members working on these projects at the moment. Those are more for the future. They're not really ready for prime time just yet. And I think one of the issues that we're already beginning to think about is how we're going to extend this out to a larger study. And one of the issues with the way we're manufacturing CAR T cells at the moment is that the throughput is relatively low, and that's one of the things that's holding us back from treating more people than we can. So we're looking into automated manufacturing systems. We're hoping to contract out some work to develop an automated manufacture process for our CAR T cell product this year, actually. Obviously, COVID's delayed a few bits and bobs of our work, and we're also in liaison with commercial companies about the possibility of commercial manufacture. Now, obviously, big commercial companies, New Zealand's not a large market, so they're tending to focus on larger countries for their manufacturing. But there are some newer systems that allow near-patient manufacturing that could emerge in the future. And so there's some other images of our current manufacturing suite. It is a licensed facility. It meets all the MedSafe requirements. It's a very manual manufacturing process, as you can see there, for example, with the cell counting. This is very good for early phase trials as you're trying to iterate the manufacturer and try to improve little things about the manufacturing process. But once we've got a stable manufacturing process, we want to try and automate it to increase our throughput. So this is the way we're manufacturing cells at the moment, and this is a good way to start for a small phase trial, and there'll still be a need for this very manual manufacturing type of process. But we're working with a company that makes some of these types of cassettes, and there's various types of cassette manufacturing where you can take large portions of the manufacturing process, put them into a closed system and automate it, and leave the cells alone for a larger fraction of the time, and that reduces operator time for various steps of the manufacturing and frees them up to do the more complex steps which we may have to do in the future. And indeed, the future could be a facility, something like this, where we actually have a large number of manufacturing pods, and we can scale it dramatically. That's probably quite a long way for our facility, but we are already beginning to take the steps of planning out what a larger scale manufacturing facility would look like, and we do have some manufacturing designs on the table for that at the moment. So we are working with a view to be able to manufacture these at larger scale in the future. So just a few concluding remarks. CAR T cells are an emerging treatment, and in some countries becoming a standard of care for relapsed or refractory B-cell non-Hodgkin lymphomas and possibly for myeloma. I would say that the large phase 3 trials haven't reported yet, so they're being licensed in countries on the basis of single-arm trials so far, so we don't know exactly who is going to benefit the most from them, but they are an emerging treatment. I think it's important that we have a foothold in this opportunity here in New Zealand. Our ENABLE trial, I think, has been really important because it's demonstrated that we can do this here. There's nothing intrinsic that prevents us in New Zealand centres, at least in our New Zealand transplant centres, of which we have several across the country, from giving this type of therapy. It's not beyond the wit of our clinicians, of our manufacturing facilities. We can do this. So we do require a lot. There's a lot we've had to put together. We've had to have systems, documentation, training, regulatory approval. It takes a lot of work, but hopefully putting this together once will make it quicker to put it together for other CAR T cell products and for other CAR T cell trials. We are still quite early in our phase 1 trial, but we're already looking ahead to what the next trial might look like. And I can't say with certainty what that will look like, but the best case scenario would be that we get good data from this first trial and we do a larger phase 2 trial. What I want then is to be able to harvest the CAR T cells at local New Zealand blood service centres across the country. And I'd like to be able to manufacture the CAR T cells and then have patients treated likely at their local transplant centres, just as they would for an autograft. That would be ideal. I don't know whether it's going to be possible. There are a number of issues around that as how we would fund a larger trial like that. And I think it will be important that we scale up our manufacture, and we're working on that automated and closed system manufacture at the moment. We're going through a few scenarios of what that type of trial might look like and how it might be feasible. And we are a little early at the moment to put in grant applications, but I think that we will be ready to do that in maybe the next 6 months or so, because I would like to think ahead to what we can do in the future. And I would say that CAR T cells are a unique product. So they fall somewhere between a bone marrow transplant procedure, which is a cell therapy, which isn't really regulated by MedSafe, and a drug. And of course, if we regulate it as a drug, then we're going to have a lot of major regulatory hurdles to be able to get our treatment across the line. If we regulate it as a transplant, the options may be a little bit more open. Clearly, we want to get the correct balance between safety and efficacy and cost. I hope we can find an avenue to this, and we are trying to speak to the Ministry of Health and MedSafe about where this type of treatment will sit and what the best options are for being able to offer this type of treatment to New Zealanders. And I think it is a nuanced question, and it's not an easy question. And I hope that we can find a way to be able to treat patients with CAR T cells such as the ones we're making, if we can show that they're safe and effective adequately. And I would just say that I'm going to go on to an acknowledgement slide in a moment. We've had a huge number of people and organisations, including LBC, and including, to give a shout out to David Downs, who've supported this trial. David, for example, has been a fantastic ambassador for the trial, and his story, you can get his book, A Mild Touch of the Cancer, which I've got here on my desk. He reports his journey through CAR T cell therapy. If you ever get a chance to see him speaking live, he's hilarious, really great, and really lovely person. And he's been quite committed, both at a personal level for individual patients and at a larger scale level, to trying to help find ways, including by supporting our own trial and our own work here, to try and make this type of treatment more available. So I think he's been a great ambassador for the study. And I do want to just finish up by thanking the huge number of people, not all of whom are listed on this slide, who've been involved in this work. A large number of people at the Malaghan Institute who've been involved helping manufacture the cell products, work our way through the regulatory settings, the financing of this type of trial, and so on. The TLR2 domain comes from collaborators at Guangzhou Institutes of Biomedicine and Health in China, and their joint company, Wellington Therapies Limited, provided some funding that helped us get the trial off the ground. Excellent team, Dr. Xin Du, who's a haematologist at Guangdong General Hospital, a very large hospital in China, and she ran an excellent trial of a very similar construct with good results, and she's been a huge help for our trial. At the hospital, we've had to go to a great deal of lengths to get this across the ground, across the line, including we've had neurologists and intensivists helping with our toxicity management protocols in case we do need those. So I just want to thank everybody on this list and a lot more besides. And I think we're going to take questions, and I'll stop the screen sharing and talk through now.
Hey, Rob, that was so wonderful. Really was. And your slides are so clearly presented. So good job. So yeah, what we'll do, we'll jump in. I'll do the talk and I'll get some questions. There's probably about 15 or so. I'll just kind of climb to the top of the Q&A box and see how you go. So one was about my lymphoma has relapsed. I've been suggested a stem cell transplant. Can I be enrolled in the Car T Cell Transplant without going through a stem cell transplant?
Yeah, so we have to be very careful. So our Car T cell therapy, it's a phase 1 trial. So we have to be very careful to make sure that people have exhausted other effective treatment options, because I cannot vouch for either the safety or effectiveness of the Car T cells. And of course, we don't yet know what the optimal dose is. So, the options vary according to type of lymphoma. So in some jurisdictions, there may be later phase trials where the safety and effectiveness is already more certain, where that risk benefit might favor going straight onto Car T cells rather than having a stem cell transplant. Our trial is, because it's a phase 1, I'm not sure that the risk benefit favors that. I would say it does depend on where you're at. So if somebody's gone into remission with chemotherapy for relapsed lymphoma and an autologous stem cell transplant is an option, has a decent chance of curing or giving a good long-term remission, it might be safer to have that procedure and then reserve Car T cell therapies for later in the disease course when we know more about how to deliver them safely and more about the risk benefit. So that might be a better option there. Generally, the patients who've been treating on this trial don't have stem cell transplant as an option. They've either been through one or potentially more than one stem cell transplant, or they couldn't go to a stem cell transplant because of no donor or no cells, or because the lymphoma never went into remission in the first place. So generally, I would recommend people have the proven treatment first and reserve a very investigational treatment like this for later. But there could be a role for that for later phase Car T cell trials.
Interesting. Thanks for that. Next one is any background on application of Car T treatment to T cell lymphomas overseas or going forward in New Zealand?
Yeah. So we don't currently have a program for that. So one of the issues with targeting T cell lymphomas is that the Car T cells are T cells. So if you target common T cell markers, the Car T cells will kill themselves. There is a group in the US, I can't remember the organization somewhere in the north of the US, that has a strategy that they are investigating for that. And their strategy is a different gene modification system where they actually delete one of the T cell markers in the Car T cells as well as inserting the CAR gene. So that the Car T cells target the marker they do not have themselves. And that can overcome this issue. So there is a prospect of that. I'm not aware of any trials on that anywhere in Australasia. I think there are trials in the United States. If you wanted to look at US trials, then that's a matter of looking at one of the international trial registries at clinicaltrials.gov, G-O-V. So there is a strategy. You have to use a different way of inserting the gene. You have to use a gene editing technology, which we're not currently using here. That has some other safety considerations. And we are looking at that type of thing for the future, but that's very much a fairly distant prospect for us at the moment. But yes, prospectively, but you have to do it in a different way.
Gotcha. Thanks for that. Next one is how widespread are side effects in treated patients?
Yeah, so I can't reveal information for our current trial yet. I would say that looking at the AXICAB, so most patients will have some episode of fever. which might indicate this cytokine release syndrome. And effectively, as the CAR T cells recognize the tumor cells, latch on and start killing them, they release the same types of cytokines your immune system would during a severe infection. And the most obvious manifestation of that is fevers. And if it gets severe, it can go along with all the things you might get with a bad sepsis, like low blood pressure. I think what's happened is that almost everybody gets some element of fever, but now we have a much better idea than we did 3 or 5 years ago about how to treat that. And people actually get a specific drug that mops up those cytokines and eradicates that fever and seems to help prevent getting more severe symptoms. So most people will get some episode of fever, and most people will get a dose of a drug called tocilizumab, which is a treatment. We actually keep a dose of it up on our ward, as well as a couple of doses in pharmacy, just in case any of our patients get that. It seems to resolve with that. So the majority of people get some sort of symptoms, usually mild, but depending on the CAR T cell construct, anywhere from 10% to 30% might get severe manifestations, and some of those might need to go to intensive care for treatment, which is why we actually have an intensive care doc on one of our committees that helps us look over and set up our policies and procedures. I don't think this is something that's going to be given in every centre just yet, and in reality it's probably something that will first be rolled out to bone marrow transplant centres, where we're used to dealing with severe toxicities and closely monitoring patients. And what I would say is, unlike allogeneic stem cell transplantation, the toxicity is usually quite short term. So it's usually, people get these problems typically in the first 5 to 10 days after CAR T cell administration, and then they resolve, and then people don't tend to have longer term problems in the same way that they do after an allogeneic stem cell transplant. That's not to say they can't, but they tend to be quite short term toxicities. So that's quite good from a clinician perspective as well as a patient perspective, because I think we sort of know what we're in for and what the period of short monitoring is. And I think that's potentially a good benefit.
Yeah, thanks for that one. Hey, the next question is, well, how many patients have been treated in New Zealand? And I think it's just a handful, but there's this other question that's kind of, 3. And then this other question is just, if things go well, what could be the full capacity in a year's time for the Malaghan Institute?
Yeah.
How many could you?
So at the moment, with our very manual manufacturing, we can really only produce about a dose a month. We have that one room that we're using. That room has to be closed periodically for a couple of weeks for cleaning. The manufacture itself takes about 3 weeks. So at the moment, we could only really produce about 12 doses a year, and that is a limitation. We have a second room so one option is that we activate a second room, but actually what we think we might do, rather than go through the rigmarole so it's not just a matter of activating a room, we have to get all sorts of certifications for it, and we have to rework the airflows and all sorts of things. And it does come at a substantial cost. What we think we might do is actually jump straight to a closed system manufacturing, which would increase the manufacturing capacity substantially. What I'd like to be able to do for a phase 2 trial is have the capacity to treat, to manufacture, say, 30 doses a year. And then if we were going to have this as a standard of care for B-cell lymphomas, the types of amount that we might need to manufacture might be more like 60 a year. Not everybody with relapsed refractory lymphoma would necessarily be the right person to treat with this treatment, because of if people have poor organ or heart function, or are very frail, it might not be the best thing. But I think at the moment about 12 a year, and we are planning. We did think about trying to double that. We're planning to actually move directly to a closed system, which I think would be a better option in the long run.
Gotcha. How interesting. So this next question is someone asking, you touched on it, but they have multiple myeloma. They haven't had chemo so far, but are there any studies of CAR T with myeloma or?
Well, there certainly are, and some of you myeloma will be aware that there are a number of studies, and both Celgene and Janssen, who are the producers of drugs like lenalidomide and Blockade, respectively, have invested in CAR T cell therapy, acknowledging this could compose part of treatment for myeloma in the future. So yes, it's eminently possible. The early response rates seem to be really good. What doesn't seem to be the case, or what we're not sure is the case yet, is whether a fraction of those patients might be cured, because most people seem to be relapsing afterwards. There's a question as to whether we're targeting the right markers on the myeloma cells at the moment. We are watching that closely, and it is an area that I think it's probably that apart from B-cell lymphomas and leukemias, it's the other strongest potential indication for now. I think that we have to be a bit conscious that we haven't got the capacity right now to run multiple or manufacture multiple CAR T cell products. So I think possibly some of the best prospects are participating in commercial CAR T cell trials in myeloma in the nearer future, where, and I agree there's a large clinical need, and it's something I'm speaking about with my myeloma treating colleagues. In fact, I'm speaking at one of their conferences, if it can go ahead, over COVID in November, I think. And I think it's a very interesting area, and we are watching it closely, and it may be something we look to for future trials. I just want to be convinced that, is there something that we can, if we're going to manufacture our own product, there has to be some advantage to our CAR T cells. So our collaborator is starting to explore a anti-myeloma CAR T cell construct, but it's not as clear to me what we can add to that scientifically, and we have to try and make sure we get as much out of the limited resources that we have.
Thank you for that. Hey, the next one is someone says, what's happening to reduce the durability of the treatment? I'm not sure if maybe it's supposed to be increase the durability. I'm not sure if I can.
So to increase the durability of the response, what we're hopeful of is that new TLR2 domain. It makes the CAR T cells, in the laboratory at least, it makes the CAR T cells more active and better able to kill a larger fraction of tumor cells. So we're hoping that that will translate to a more durable response. That's the scientific hypothesis around the construct. The trial mainly is that whenever you add anything new to a CAR T cell construct, the risk is you might increase the toxicity. And so the trial is a safety trial primarily to make sure we're not getting undue high levels of toxicities. And that's why we're starting at a low dose and working our way up with this construct. But that's the rationale of what we would hope for. Yeah.
Okay, I appreciate that. This other question, and I know you touched on, so this is T cell therapy an option for T cell lymphoma, in particular mycosis fungoides?
Yeah, so as I say, might be an option in the future. As far as I know, most of the trials have been more in peripheral T cell lymphomas rather than mycosis fungoides. So it's a potential prospect, but it does require more complex gene editing. And not actually even necessarily more complex, newer and less proven techniques in the laboratory manufacture of gene editing techniques, which is not what we're using for this current trial.
Appreciate that. And this other question is, does this have potential for acute myeloid, AML?
Yes, it does. And in fact, the first CAR T cell trial in Australasia was in AML. And it was run by my predecessor here at the Malaghan Institute called Dave Ritchie, who's based now at the Peter MacCallum Cancer Centre, and in fact now chairs our Data Safety Monitoring Committee. Yes, there's a lot of interest in using CAR T cells for acute myeloid leukemia. The problem in AML has been identifying a marker on the AML cells that is not shared by normal cells that make neutrophils, or there's normal stem cells that make neutrophils. And that's been the great challenge with AML. So one of the interesting things about B-cell malignancies is there are a lot of different markers on the surface of B cells that aren't really expressed on any other cell type. And you can live without your B cells. You might need to have immune globulin infusions to help prevent infections, but you can live without B cells. On the other hand, you can't live indefinitely without neutrophils, without normal neutrophils to fight bacterial infections. Obviously, we can for perhaps weeks or even months at a time with appropriate antibiotics, but it's necessary that we have neutrophils. And one of the problems with targeting AML is that these treatments can be so effective, you can eradicate all the bone marrow and then have no ability to manufacture neutrophils. And the CAR T cells may also eradicate, if you try and do a bone marrow transplant, they may also eradicate those stem cells as well. So it's been a big challenge. There's a lot of work going on internationally. I think there are issues around the what to target, and yeah, we'll just have to keep watching that closely. We can't answer all the questions in a unit of our size, so we have to pick and choose answers. But there's so much research work going on into this globally, and it's such a modular technology and modular technique that I think we will see other indications coming through in the future.
Oh, very interesting. Thank you for that. This other question is, yeah, we would like to find out more about immunotherapy treatments that are available for lymphoma treatment around the world, and if they are being used here in New Zealand, and if not, why not?
Yeah. Well, I mean, we are, just saying, immunotherapy is quite a broad term, and in a way, allogeneic bone marrow transplant is immunotherapy. Also, the drugs such as rituximab or obinutuzumab that are used to treat lymphomas and B-cell leukemias are really immune therapies. They're antibodies that latch onto the tumor cell and label them for the destruction. There's a question in there I can see particularly about pembrolizumab. So there's this type of immunotherapy called checkpoint blockade, which unleashes T-cell responses against tumors. It is funded in New Zealand for melanoma, and I'm aware that there is currently an application, which I was partly involved in helping with, to try and get that funded for a type of lymphoma called Hodgkin lymphoma. And Hodgkin lymphoma doesn't respond to our type of CAR T cells. They're only really for non-Hodgkin lymphomas. So yes, there is some work to try and get those available here. I believe that some people have accessed some of those immunotherapies either on compassionate access schemes or by self-funding or by named patient applications to Pharmac for short-term use before a bone marrow transplant. And there's also a question there about those checkpoint blockade medicines, giving them after CAR T cell infusions. Yes, so this one type of immunotherapy called checkpoint blockade works by unleashing T cell activity. And there have been a few case reports where people have been given CAR T cell therapies, their lymphoma has relapsed, and they've then received this checkpoint blockade, and that's reactivated the CAR T cells, and the lymphoma's gone back into remission. Those are fairly sporadic reports so far. We haven't done that in our trial. I'd be a bit worried that we don't know the toxicity of our CAR T cells yet, and we might just increase the risk of toxicity, and that might prevent us finding the right dose. So I'm a little bit nervous about doing that with our highly investigational CAR T cells. But yes, you can combine CAR T cells with other immunotherapies potentially. That's right. I just would also give one shout out, which I think is an important point to make, is that CAR T cells are one way of activating T cells directly against lymphoma cells. There's another related approach, and even though ours is in Wellington is the only CAR T cell trial in New Zealand, there is a related approach called the BiTEs, which are antibodies that latch onto the lymphoma cell on one side and latch onto T cells on the other side. And the advantage there is you don't have to manufacture a personalized cell product. You can't quite activate the T cells in exactly the same way as you can with CAR T cells, but there's very promising new BiTE technologies. And there is a BiTE trial for certain types of lymphoma operating in Auckland. And we have suggested it to a couple of clinicians who nominated potential participants for our CAR T cell trial, but who weren't able to be treated on it. So there are other approaches, and I think the more of this type of research and work there is, the better, because that will find the best way of using these treatments.
Thanks. Good to hear about the BiTE molecules. Yeah.
There is a question, sorry, I just mentioned there's a question about Burkitt lymphoma, and I think it's been self-answered. Burkitt lymphoma should, in theory, respond to our type of anti-CD19 CAR T cell lymphomas. The problem with Burkitt lymphoma is it's characteristically an extremely aggressive malignancy and progresses very rapidly. And at the moment, we are still, one of the purposes of our trial is sorting out the feasibility of manufacturing. And we're still working through ways to accelerate and make our manufacturing processes more reliable. And our concern about Burkitt being such a very rapidly progressive malignancy is that it would simply not be feasible to manufacture the CAR T cells and deliver them in time in an early phase trial like this. So that's the rationale for our concern about including Burkitt. So I don't think we can treat patients fast enough in this very early phase trial yet. Some units are working on accelerated ways to manufacture CAR T cells faster, and there's also a field of work in whether you can make CAR T cells from other healthy donors and give them to patients, that type of thing might be more feasible because you could have the CAR T cells ready off the shelf and give them at short notice. That might be more feasible for Burkitt lymphoma.
Yeah, thanks for catching that one. Hey, just before, I want to make sure it's in the chat box too. Somebody was asking about Waldenstrom's.
Yeah.
You know, is there any potential, for CAR T?
Is a potential cure cure. Yes. Yes, there is. I'm not aware, there's not a great deal of data on CAR T cell therapy for Waldenstrom's at the moment. There is an emerging bit of data for marginal zone lymphomas, which are most similar to Waldenstrom's. One of the issues with CAR T cell therapies is that having a lot of disease in the bone marrow can be linked with a higher rate of severe toxicities, and Waldenstrom's often has quite heavy bone marrow involvement. I guess I would say that the biggest gap in our treatment offerings for Waldenstrom's at the moment in New Zealand is probably BTK inhibitors, such as ibrutinib or acalabrutinib or zanubrutinib. And I think that an important treatment to offer for Waldenstrom's here is a treatment such as that. And I know that companies are beginning to put in applications to pharma for that. And if I were a patient with Waldenstrom's lobbying for treatments, I think BTK inhibitors are a really important treatment for that. Similarly, we're not currently treating patients with CLL in this trial because we think that all the disease in the blood carries the risk of severe toxicity. And again, probably the biggest priority there is getting BTK inhibitors, or at least one BTK inhibitor funded in this country. But look, we're keeping these things under review. It's very difficult for us to treat every disease.
Appreciate that. Hey, Rob, going back to the Q&A, there's a few kind of lab questions. How many groups of 3 will you need to confirm safety?
Well, we treat, what happens in this trial design, it's called a 3+3 design. You treat 3 people at one dose, and then you escalate a dose. You treat 3 people and escalate. If we get one, and we have a particular definition of toxicities that would stop us going any higher. So if we have 2 people out of 3 get any of those bad toxicities, we stop at that point, and we have to say that that dose was too high and step down a dose. If we have 1 toxicity out of 3 at a particular group, we would say that we don't know. We treat 3 more at that particular dose to assess if that dose is indeed has acceptable safety or not. So we have a schema about that, and how many we have to treat, we don't know. We've estimated that the most likely number is around 12 in this trial, but it really does depend. If the next 2 patients had a severe toxicity, we might only treat a total of 5 patients in the trial. We don't know. What we may end up doing, particularly if we make significant manufacturing changes, is we may apply to run an extension study where we treat more patients at the defined dose, having made some manufacturing improvements. So we might yet make a modification, but we'd have to get ethical approval for that, of course, and we have to be clear that there's a clear rationale for it as well, scientifically and clinically.
How interesting. And another lab question what is it like to work in your manufacturing lab or in the highly secure labs? We all got used to it. Yeah.
Okay, so the first thing I have to say is I have never set foot in that lab. Actually, that's not true. I haven't set foot in that lab since it was established for CAR T cell manufacturing. I've set foot in the lab, but not since. And the reason is it's incredibly access controlled. So all our GMP manufacturing team have to have extensive training in gowning and degowning. There's even a standard operating procedure for opening and closing the door, and that's to check that the airflow couldn't let contaminants through. So, but I am reliably told wearing all that garb is quite horrible, and it is quite sweaty. It's air conditioned inside there, but and temperature controlled, of course. But it is awkward wearing all that. They have to wear 2 pairs of gloves, so double glove inside the room, and so it is pretty hot and horrible. Even with those closed manufacturing systems, there will still be manual manufacturing steps the initial cell processing, and then decanting the cells at the end into the vials to store them, are likely to remain manual for the foreseeable future. But it will reduce significantly the amount of time going in and out of that room, which will be a lot easier for the operators. So yeah, it's not very nice, and a huge credit to the team, Brigitte, Reggie, Kirsty, who have been doing great work in there.
Appreciate that. I'll just, yeah, no, we're getting towards the end here. One question here is, are there certain characteristics with people that fail to respond to CAR T cell therapy?
Not actually even so much patient characteristics, but if the cells don't proliferate well inside the patient, then it usually indicates that there won't be a response. So it seems that successful CAR T cells actually proliferate inside the patient. Again, the pharmacokinetics of these are completely different from a drug where you give it and it gradually declines. You give the CAR T cells and it increases a few days later and then declines again. So it's quite a different scenario. Generally speaking, most of the disease characteristics that predict poor response to chemotherapy don't predict a poor response to CAR T cells. There's a tendency to people with very bulky disease to not get into a complete remission, and this is based on the licensed products and the largest trials of those so far. But yeah, primarily it seems to be, it may be that there's something particular about some people's CAR T cells that the T cells are not very fit, and they're not very able to proliferate very well, and therefore to kill very well. So I'm not sure that it's something down to the patient themselves. There is a bit of concern that the accumulated effects of lots of prior therapies may affect the T cell fitness, and some units with more established CAR T cell therapies looking at getting them earlier in treatment, but we don't know the results of that yet.
And this other question is kind of related. It just said, they said, if a person has CAR T and subsequently relapses, can they further have more CAR T down the
Yeah, so probably not the same type. So if you're giving a CAR T cell of the same type, they tend not to proliferate very well, and they're probably resistant. But one of the areas that we might look at for a next trial is CAR T cells that target more than one antigen on the B cell. And so, yes, there's a possibility that if you don't respond, and there have been a few case reports of people who didn't respond to a CAR T cell directed against one marker on the lymphoma cell, say CD19, and then did respond to a CAR T cell therapy directed against CD22, which is another marker on B cells. So there is the potential, but it wouldn't be rational to give the same CAR T cell product again, because we wouldn't expect that to work. One thing we're looking at is whether it might be a good thing to look at for a next trial as a CAR T cell therapy that's directed against more than one antigen. Then it could be an option for people who failed another CAR T cell therapy, as well as people first up. So that might widen the applicability of the treatment. So we are actively looking at that.
That would be interesting, actually.
As a question about ibrutinib, ibrutinib is not funded for Waldenstrom's, but it is for all of the non-Hodgkin lymphomas. Do you know why? It's not funded for all of the non-Hodgkin lymphomas. It's not funded for any indication in New Zealand yet. It's got a license for CLL, and it's not funded for any indication. And personally, I don't mind whether it's ibrutinib or acalabrutinib or zanubrutinib. There's 3 BTK inhibitors now around. Not all of them. Only one of them licensed in New Zealand, but we really need a BTK inhibitor in New Zealand. And Waldenstrom's and CLL, I would say, are the priorities. Yeah.
Yeah, appreciate you grabbing that one, Rob. Yeah. Hey, this one came to me from a patient. They saw last month FDA approved. I think it's called Tiscardis. It was the mantle cell lymphoma, like for CAR T. Did you see that?
Yeah.
And so just.
It might be tisogene-let-lucel. I'm not sure. Maybe it's another one of that. I have to double check.
Okay.
There've been some good data. So we do specifically include mantle cell lymphoma in our trial because the emerging data from mantle cell lymphoma responses to CAR T cell therapy are good, and there are good data for Axicab, which is the one of the other CAR T cell therapies as well for mantle cell lymphoma. So yes, I think. That is a lymphoma that does seem to respond well to this type of treatment, or can respond well to this type of treatment. Whether our therapy is the right one, I don't know, but it's certainly an option after you've exhausted other good treatment options. Yeah.
Okay. Thanks for that. Hey, just
Great.
this might be my last question. Just somebody was just asking if a patient goes into Car T, like they go into the procedure with like a relapsed disease. Is there like a difference from someone who maybe can't go into an allogeneic because they couldn't get into a?
So what we're just requiring from an ethical perspective is that people have exhausted other treatment options. So if they can't go onto a transplant because they haven't reached a deep enough remission, we call that refractory disease, and then that could potentially be an area where CAR T cell therapy becomes useful. Yes. So it's not that you have to have had an autologous or an allogeneic transplant. We just have to have exhausted, other viable treatment options. So yes, if you couldn't, so it's not that we're requiring everybody who's had a transplant procedure. We just have to know it's been considered and that conventional treatments have been exhausted first.
Okay.
And if it's been declined for some practical reason, or or because the depth of response from prior therapies wasn't deep enough, then that might be reason to consider CAR T cell therapy. Yes.
Okay. All right. Hey, I appreciate it, Rob. I might just have to call a wrap, and because you got to get some lunch, and we're really working you. Hey, just wanted, but on behalf of us, LBC staff here, and just on behalf of the patients and families around the country, thank you. Thank you for just giving of your time today to do a webinar series with us. So.
That's all right. Well, thank you very much, and thank you to LBC for supporting this important work.
We appreciate it.
Thanks so much.
Yeah, take care. Go well.
That's great. Okay,
right.
Thank you. You too.
Thanks,
Stay safe, everyone. Bye-bye.
Bye.
Bye.